Perfect. Nanda, you can start.
Yeah. Hi everyone. I'm Nanda Kishore, the workshop chair at ISDAIOT Symposium. So hope you are all keeping well. You might remember me from the data analytics workshop days. So this is today's session is sort of like going to be a sort of continuation, not exactly a continuation because obviously we had a decent gap in between, and also it's not um, exactly a continuation of that in terms of topic as well. But uh, what we will have today is a brief discussion and introduction into bioinformatics and how you can sort of analyze the apply data analytics tools to the field of bioinformatics. So which is more like a sort of, we learned the Python and related topics in the data analytics sessions. So today you'll be exploring more like more from the practical application perspective. So before I begin with the session, just some usual housekeeping stuff. So this is going to be around a 45 minutes talk followed by Q&A. So please use the Q&A window to type your questions during the session. You may also raise your virtual hand from the bottom tab at the end of the session so that we can unmute you if you like to ask any questions directly. So as I mentioned, today's talk is titled Bioinformatics and Python for Untangling Genomes. That's being delivered by Dr. Sanjay Patil. So Dr. Sanjay Patil recently completed his PhD from the School of Physics at the University of Melbourne. So during this PhD, he mainly worked in observational cosmology group in the physics department. While his main research area is astrophysics, he also works in several interdisciplinary areas like bioinformatics, data analytics, and machine learning. During his PhD tenure, he has delivered more than a dozen of research talks at global venues, ranging from Australian National University in Canberra to Stanford University in USA. He has received various honors and awards like Science Student Ambassador, John Lady PhD Travel Bursary, Melbourne International Engagement Award, and Thesis Write-Up Awards. So what he'll be discussing today is mostly he will be following the format that we followed during our data analytics workshop, which is a combination of oral presentation followed by a bit of Python coding using the Jupyter Notebook. So I hope you all have fun, and I hope you get to know this is the last session in the symposium. So I hope you enjoy the session. So Sanjay, the session is yours.
Thanks for the excellent introduction, Nanda. First of all, I would like to thank the organizers for giving me the opportunity, and I would like to thank you for all joining, and congratulations for all those who just finished their exams. So this is a part of workshop series and data workshop series which started in mid or end October. And today is the last workshop of the series, which is named Bioinformatics and Python to Entangle Genomes. So in the previous workshops, you have seen, if you have attended, you have seen fundamentals of data handling, how to read data structure, how to visualize it, how to explore and data processing techniques, and some machine learning algorithms as well. So if you have not done it, this session wouldn't be much harder. So I think you can still follow. I would like to keep it interactive. So if you have any questions during the session, please feel free to ask it in between. So the last session, in this one, we are going to look at what the bioinformatics field is as such and see how we can use Python to analyze the data in bioinformatics. Without further ado, I will start. Oh yeah, as Nanda mentioned, I recently finished my PhD earlier this year. So I did work in astrophysics. So I'm thinking now in the transitionary phase, I'm looking into options, what options I have, what other fields I can look into. And one of the exciting and interesting fields I came across is bioinformatics. What exactly is bioinformatics? It's a multifaceted discipline. It includes biology, computer science, physics, mathematics, statistics, and genetics, and obviously data science and data analytics people. So it's a way of looking into the biological problem from a computational point of view. So it involves essentially developing software tools, mathematical and statistical techniques to robustly analyze the biological data. Why do we need to do bioinformatics? You guys might have wondered, like, what is the difference between an individual who is shorter or an individual who is taller? What is the difference, a basic difference between these two guys? So if you look at the very basic difference, it is in form of a genetic difference, which is of the order of 0.1%. It's a very little difference, and it is encoded as genetic information. And this genetic information essentially acts as a manual to produce molecular structures, which in turn produce biochemical functions, which in turn leads to characteristics of an organism, such as height, color, or even the propensity for a particular individual to have a particular kind of cancer. So it's essentially by developing bioinformatic techniques, we are developing techniques for analyzing this genetic data and related structures. We are attempting to understand the basis of life at a very molecular level. So bioinformatics is interesting and exciting. At the same time, it has numerous applications, right? To name a few, so it can be used in personalized medicine, it can be used in gene therapy, it can be used in crop improvement, it can be used in climate change, etc., etc. Like many other fields nowadays, bioinformatics is also inundated with huge amounts of data. So the data has been increasing exponentially. If you look at the plot on the right, just ignore what has been written on the y-axis. Y-axis basically says the amount of data, which I will come back to the sequences later, what sequence exactly mean. It's essentially, you can just assume it for now as amount of data. So the amount of data, and on the y-axis is the time. So the amount of data is increasing exponentially over the years. And nowadays it's taking even less than a year to double the amount of data. And in order to fully realize the potential of this huge data sets, you need to effectively analyze the available data sets. You need to develop techniques and tools to analyze the data sets. That's where bioinformatics comes. The goal of bioinformatics can be categorized into three main branches. One is data curation, other is data developing software tools, and the last one is the data analysis, right? Data curation is essentially the way to store data, the way to retrieve data. It involves data structure. It involves efficiently retrieving data and storing data in a particular format. And the second one is developing software tools. It involves algorithm development and other development to efficiently analyze the huge data sets of bioinformatics or biological data sets. And the last one is the data analysis, where we try to extract information, biologically meaningful information from the available data sets that we already have. For example, if we have the genomic data from say human and a monkey or a chimpanzee, so then we cross-correlated those two data structures to see how the evolution actually is happening, right? So there are quite a few interesting things, and in this particular workshop I'm going to mainly deal with some intersection between the last two, that is software tool development and data analysis. I essentially look at a few algorithms to how efficiently we can analyze the biological data sets we have, right? So before going into the algorithms and how we analyze the data sets, I think it's important to have a brief understanding or have a basic understanding of what data set are we actually dealing here. So the data sets which I mentioned earlier, it's the genetic information, is a recipe of an organism. It says how the organism is going to evolve. So if we have the recipe and we can simulate the organisms essentially, right? And the basic unit is the DNA. So DNA is this double helix string made up of basic units called nucleotides. And each nucleotide is made up of mainly three components. One is nitrogenous base, second one is deoxyribose sugar molecule, and a phosphate molecule. So approximately, a nucleotide can be categorized into four different types. Based on the nitrogenous bases, because the deoxyribose sugar molecule and the phosphate molecule are essentially the same. So the four different nitrogenous bases are adenine, thymine, guanine, and cytosine. This can be abbreviated as ATGC. So a genomic data, if you look at a genomic data, it's nothing but a string of 4 letters, and it can be a bit on first order approximation, it can be divided into 4 letters, ATGC. So the example here, which I have given on the last one, is an example of genomic data taken from the Vibrio cholerae bacterium. So it's a huge data. I just took a sequence. So it appears as ATC, AA. So it's a string of data essentially, and these are huge. For example, the human genome is itself made up of 3.2 billion nucleotides. It's that big, right? And we need to extract meaningful information from these ones, we need certain techniques to analyze this huge data structures efficiently. As I said, Python can be used for bioinformatics. There are quite a lot of Python-related open source softwares where you use Python tools to analyze the data sets. And I'm just listing few here. So important ones are BioPython and BioTite. And these are open source softwares which use NumPy arrays and other data structures that are available in Python to efficiently analyze the data sets, biological data sets we have. So I recommend everyone to go through this open source softwares and just get an idea of how to use them and use it at your own free time. Other ones are the Flash and Blast. These are algorithms which can be used to analyze the biological data sets, right? I don't get into the details of it today, but I just encourage everyone to have a look, to go and have a look at your own free time. So today I will be mainly discussing the algorithms. I will in the next part of the workshop, I will be mainly discussing the algorithms which are used to analyze these biological data sets. An algorithm is nothing but precisely defined set of steps to solve a problem of interest. And we need efficient algorithms to analyze the bioinformatics data, right? I will first go to the Jupyter notebooks that I have created. If anyone has any question, you can stop me now, and if you ask, or if not, I will just jump to Jupyter notebooks where I will show how to develop very basic algorithms to analyze data.
I think we'll take the questions in the last, Sanjay.
Right.
Probably if they send in chat, I'll stop you.
Sure,
do you want me to share the notebooks with everyone?
Yes, please. Karishma will share notebooks now. So if you have facilities to access the notebooks, and we can code along with you, but if not, I will just code and you can just look and understand, and you can do it later. Right, the first one I will be looking into is the frequency counts or the pattern counts. So if you have, so is the Jupyter notebook visible for everyone? Let me see the chats. Cool.
Yeah. We can see the notebook.
So, sure. The first algorithm I will be talking about is finding replication origin, right? As I mentioned earlier, genetic material or the DNA is nothing but the recipe for an organism. So the quantity of genetic information in any given cell of a human being is same, right? So when the cell divides, the genomic material has to replicate itself, right? And the area where as I said, this human genome is 3.2 billion nucleotides long. So there has to be a specific area within, or the region within that huge genomic sequence where the replication starts. And this area where the replication starts is called the replication origin. So the replication origin is mediated by a DNA protein that binds to the short segment within the origin region or within that particular sequence. And in layman terms, DNA box can be thought of as a particular sequential arrangement of nucleotides, which is in the DNA specific language and which can be translated loosely into English as bind here. So that DNA particular A sequence is what tells the DNA A box, which is the protein, to come and attach the genomic sequence there. So it's of biological importance to look for frequently repeated nucleotide sequences in the origin region. In this workshop, I will explain a few algorithms for their implementation. For example, so the algorithmic question here is you have given a genomic sequence, which is this one. So this genomic sequence is it's just nothing but a string of letters A, C, A, C, T, etc., etc. And we need to develop an algorithm to look how many times this particular pattern, which is AC, TAT, has been repeated, right? So the output, we can see that the output is 3, but how to develop an algorithm, right? So the way to develop, it's the very simplest one. What we are going to do is we are going to define a function, right? Pattern count is the function, and we are going to pass in the genome sequence as well as the pattern, right? And what it will do is it will start from the first one. It will start from here and go for the length of the pattern, right? It will go 1, 2, 3, 4, 5. So the 5 one, and see whether this pattern matches this sequence matches the pattern that has been given. If it is not, then it won't add to the count variable, right? Or it won't add to the frequency of variable. So next, it will go to the second one, and it will again read the next 5 letters of the sequence. And if that matches with the pattern, then it will add again, right? So we need to initialize count equal to 0. Right? And it will go from I equal to 0. If you have attended the previous ones, you know that the indexing in Python starts from 0, right? So it starts from the 0th element, and it goes up to the length of genomic sequence minus the length of pattern. Because all we need to do is we need to go up to here. We can't go here because there are no 5 elements, 5 letters after T. So you have to go up till A. So you have to go from 0, you have to go till length of the genomic sequence minus the length of the pattern, right? And you see whether if you put a condition to see whether the sequence of letters match the pattern. Right? If genome sequence of I is 2I plus K. So it is taking all the sequences, all the 5 letters from here, from the starting, and see whether it matches the pattern. So if it does, then it is going to add to count equal to count plus 1. And then finally it returns. So this is the algorithm basically. So we are going to implement that particular algorithm in Python. So we will define a function. Function is defined by def is the root keyword here. Def pattern count, and then include genome sequence pattern count 0 for I in range of length of genome sequence minus the length of pattern. So this is the for loop in Python, which is equivalent of this particular algorithmic step. So it goes from 0 to the length of genome minus the length of pattern. So it will transfers through the slide. It slides through the genomic sequence. And now the if condition. If genome sequence I is to I plus length of pattern. So you're going from that particular 0th index to the index up to 4th one or 5th one, sorry. If that holds, then you are going to increase the count by count plus 1 and you will return the count. So to run this, all you do is shift and enter. So now this function has been already incorporated in the Jupyter notebook. So I'm showing you an example. One of the examples is this given particular genome, which is ACAA stuff. And you need to see how many times this particular pattern has appeared in this genomic sequence. So all you do is call the function, call the function pattern count, send the genome, first one, and send the pattern. This is the pattern. And if you want, you can see this. Now I have to print output, which I want. It is showing me 25 times. Let me see where I went wrong. Oh, sorry. So I have to see whether it matches the pattern actually. Yeah. So I have to see whether it matches the pattern, and if it matches, only then I will have to add it, right? So when added, so this particular pattern, which is ACTAT, has been, you can see it here, 1, you can see it here, 2, and you can see it here 3 times. It has repeated a total of 3 times. But you can change the pattern as well. So if you want to see whether there is this pattern, how many times this pattern has been repeated, there is only 1. So only this carries this particular pattern, right? So you can also read the genome, which is in a file, because sometimes the genomic sequences are huge. You can't have it as a string. So if you can have a file, so probably Karishma has sent you a file which has dataset pattern.pkl.gz. You can also, this is a .pkl.gz version file, is a Python related files. So I have they have stored the genome and the pattern they need in this particular file. So we can open the file. To load the file, so we need to import pickle and gzip. So data is equal to pickle dot load gzip to open 1. So you have loaded the file and see what format is the file, like what kind of data structure have you loaded. To see what kind of data structure, you can just say type and data. And if you shift and enter, okay, there is a problem here. Gzip open. Okay, so we need to do R. So we are reading a letter. So print type. Okay, there is no such. It's giving me an So we are loading the data and seeing. So when there is an error which says dataset pattern PKL. It says there is no such file. So you need to make sure whether you have the file in the directory that you are actually running this Jupyter notebook. So it's saying that the error that I'm getting is it's saying I don't have such files in Jupyter. Let me just quickly check. Oh, so yes, sorry. So I have written it as data.pattern. So yeah, so make sure that you give it the right name. So if you look at it, the type you get is dictionary. So data, the data structure is dictionary type, right? So you can print what keys it has. So it has two keys. One is genomic sequence and another is pattern. So you need to see how many times this particular pattern has repeated in this genomic sequence. And to do that, all you do is call the pattern count and give it data of genomic sequence and data of pattern. So you send it these two arguments for this particular function, and that will be output for this one. And you can print. Yep, for that particular one, for that particular genome sequence, and we can see what the genome sequence is. So genome sequence is a huge one, and it has the pattern is. So this is the genomic sequence that we are looking in, and this is the pattern that we are looking. So this pattern has repeated in this particular one 23 times, right? So I hope that this particular algorithm is clear for most of you. Now I will be moving into the next algorithm. This is in the links that she has sent. So you must have found frequency count. So just open this particular one. So this algorithm mainly looks for how many times a string of particular length has been repeated. Say you want to see a generated sequence of 4 string length, like ATGC. So how many times how frequently has any particular given sequence has been repeated that many times, right? Say I will just take the same one. Probably this will explain much. Say what you give is genome sequence and length. Say 4. Output should be how many times each sequence of length 4 has been repeated. What I mean is, say you will look into ACAA, how many times this has been repeated in the whole string, or you look into CTAT, and also it will return how many times this particular one has been repeated and how many times this particular one has been repeated. So we need to write an algorithm which will look into the string length of 4 and see how many times each string has been repeated, right? So how do you write an algorithm for this? So there is one easy way, which is going to be involve huge time. So it's going to take a lot of time, but it's an easy algorithm. So I will just define that algorithm first. I will discuss that algorithm first. And then after that, I will go to another algorithm, which is much more robust, and it will perform the same job at a fraction of time the first algorithm uses. So call it as a function frequent words. And what you give is two arguments. One is genomic sequence, and the other one is the length, which is the key. K is the length. K is the length, and what we are going to do is we are going to initiate frequent words. Probably it might confuse you guys. So frequent pattern. I'm going to initialize an empty set or empty list and count, which is also an empty list. And I'm going to call the pattern counts that we have defined earlier. Length of genomic sequence minus k, which is the length of the string. So the idea here is you slide through this. So you go from here first, I equal to 0, and you take the first 4 elements and see how many times the first 4 elements has been repeated in this whole genome. And then you go to the next one, which is CAAT or CAAC. And then you're going to see how many times this particular string has been repeated in the entire genome. And then you will go to A and so on and so forth, right? So this for loop essentially does that. So this for loop goes from here to here and see how many times that pattern, the first pattern is given by genomic sequence. Okay, right? So this is the pattern, this is the first pattern, and you're going to count that pattern in the genomic sense. For that one, you can just use this function, right? You have already written pattern counts. So what we do is we are just going to go call the pattern count, genomic sequence, comma, pattern. Okay. That essentially is temp count and count of I equal to. So basically count of I, it's a list which is going to say the ith position that this particular string has been repeated this many number of times, right? So this will have a list and it will have the number of times each of the possible sequence has been repeated. So you need to calculate the max count, how many times that maximum it has been repeated. So it will be max of count. So you will count the max list. And then once you have it, so you're going to, we're again going to slide through it, right? So you're going to see which pattern corresponds to the max count, right? So you're going to through the string from I equal to sliding through the genomic sequence, length of okay. And if the count of I equal to max count, right, then you're going to add, and then you're going to add that particular pattern, which is okay to frequent pattern. And then you need to remove the duplicates. So now we have the most repeated sequences in the frequent pattern list or the data structure. Now we need to remove, because there might be a few strings which have been repeated. For example, if the for length is done, so then there will be ACT80, and this one is also called ACT80, but so we need to remove the duplicates. So in Python, there is a very easier way to remove the duplicates. All you need to do is return set and whatever the list is, just pass in the list. So set basically removes all the duplicates and it will give the only unique ones, right? Right, that's the algorithm, how to implement it in Python. We will define the function, which is frequent words, and it will pass in two arguments. I will just copy and paste, right? And we will use the frequent and count to lists, empty lists. We are initiating empty list, and for I equal to 0, I in range of length of k. That is the thing. Then you are going to get pattern. So some of the algorithms you can just copy, because it's in Python, it's very easy to code. So using the pattern count and then max count equal to max. And again, you're going to slide through the genomic sequence again. So I'm going to copy. This is the for loop which does the job, which slides through the genomic sequence. And if that particular count has equal to the max count, then you are going to add the pattern. Make sure the indentation is correct. The particular pattern is appended to the frequent pattern one. Right? If you run, right? So it just runs. Circle. Now I want to see how many times. So we get an error. So the error is, okay, so we defined the pattern count in the previous Jupyter notebook, but we don't have that particular function here. So all we need to do is just copy the one in the previous one, and just do this. So now it knows, because this particular Jupyter notebook has no idea what actually pattern count is. So we need to define it here as well, right? So when you do that, and then you do it. Yeah, so there is another one. Okay, so because you can't have in this one, because you initiated it to a negative, empty list. So all you need to do is instead append is the one which appends every time you count the pattern. And then you do. There is a spelling error. Yeah, there's stupid errors I do all the time. Yeah. Okay, so now, so it shows like how many times this has repeated. So there are repetitions here. As you can see, GCAT has been appeared 3 times, and CATG has also appeared 3 times. Because what I didn't do is I didn't remove the duplicates. To remove the duplicates in Python, it's very easy. All you do is set, which is a function which removes the duplicates. If you okay, let me see. Yeah, there you go. So what it says is that this 4-letter string have been repeated the most number of times, right? Okay, so if we have time, I can go through another, because this particular algorithm has so many for loops which have been embedded in them. So for loops are notoriously time consuming. So if you have 2 for loops, it's going to take on order of n square. So the time consumed to go through that algorithm is exponentially increases as the length of the genomic sequence increases. So if we have time, I can go through another one which executes the same algorithm and which does return the same output, but in a very fraction of time compared to the first one, right? So the algorithm basically uses the hash table. Hash table is the kind of data structure which to look into that data structure, it doesn't depend on the length of the data structure. Say it's a data structure containing n elements, and you want to look into whether that particular element is present in it. So if you search in it, it doesn't take much time. And the hash table, it depends on from one programming language to another. So hash table for example, in Python is dictionary, right? So the idea here in this particular algorithm is the earlier algorithm has too much time, like it takes too much time. It will reduce the time complexity of the earlier algorithm by trading space for speed. As I mentioned, and embedded loops are notoriously time consuming. So this particular algorithm, what it does, it essentially goes in the same way, right? Frequent words. I will just do optimal to say it's an optimal one. I think you can do much more optimal, but for this particular workshop, I'm just going to show one of the ways to make it optimal. So you will do frequent pattern equal to initiated to empty hash table. Right? And then go through the sequence for I. From 0 to length. Right? The idea here is you go from, similarly as you traversed in the previous algorithm, you go from here to here and see whether this entry has been already made in the hash table, right? And if the entry has been already made in the hash table, then you are going to just add one more to the count. So what you are going to do is, so the pattern here you have is the genomic sequence of I is to I plus k, right? And if the pattern exists in this hash table, that is the frequent pattern, if it exists, then what you do is frequent pattern, you increase the hash table for that particular pattern by 1 equal to frequent pattern plus 1, right? If not, which is the case when it goes for the first time, when there is first time the hash table is empty, so there is no entries in the hash table. If there are no entries, it's going to return 0. So then you need to initialize that particular one to 1. Right? All you do is return frequent pattern. So essentially, we reduced earlier one has a for loop here, And the pattern count, if you remember, has a for loop as well. So there has 2 embedded for loops and there is 1 more for loop here. So we are going to reduce 3 for loops just for 1. So it's going to improve the performance, the time for the performance considerably. So how do we implement in Python? Python 1, all you do is you define the function, frequent words, right? And frequent pattern, as I mentioned, this is an empty dictionary, which is the hash table. Initiate an empty dictionary, right? And go through the string. To go through the string, this is the for loop. We have seen it quite a few times now, right? This is the for loop. And you check whether that pattern, you check what pattern it has. So this is the pattern, and see whether the pattern exists in the particular keys. If so, all you do is else you're going to initiate. Right? Okay, so we have now run it. So I'm going to use the same example and let's see whether it will give us the same result. It should. So frequent words. Let's see. Oh, there is a mistake. Okay, so there is again a spelling mistake. So you need to make sure that if you initiate it as a whatever spelling you use, so it should be exactly the same pattern. And this should be pattern. There's again an error. Okay, sorry. So I think there is one more here. I think it would have been better to change. Yep. So you will get a dictionary of how many times this 4 string has been repeated in this particular one, right? So if you look at it, so this is a dictionary and then it's clearly 3 is the maximum and 3 has been repeated twice. So one is GCAT and CTAG. So this has been, if you go through this one, which is the first one, yeah, so C tag and this one. So yeah, we got the same answer, but for a fraction of time, which it took to run for the first algorithm. So that's pretty much the algorithm part of it. And we almost came to the end of the presentation. So to conclude, so genetic data is, by studying genetic data, we are looking life at a very basic molecular level, and it has, by doing so, we are have numerous applications with bioinformatics, and bioinformatics current days is being inundated with data, and robust data analysis is the key. For robust data analysis, we need to have efficient algorithms, and in this workshop, we looked into few algorithm developments. So that's pretty much all I have got to say.
Thanks, Sanjay. That was a very interesting talk. If you have any questions, please feel free to go ahead. Will there be a recording of this provided? Yes, there will be. The recording will be made available in the YouTube channel. So.
Yeah, and the links of the Jupyter notebooks have been also made available, so you can have a look as well.
So we have a question that says, can you please discuss further on hash tables?
Well, so hash table are kind of data structures. It depends. It varies from programming language to programming language. So hash tables are this data structures where if you want to search for a particular entry in that data structure, so it doesn't take much time. So it takes an order of one time. So it's a simple. It doesn't depend on the length. So if there is a huge data and you want to search, and if it is an array or something, then it takes more time to look for it. But if it is a hash table, which is in the case of Python, dictionaries are considered as the hash tables. So it essentially it doesn't take time at all. So by that way, we are going to increase the efficiency or decrease the time that it takes to run the program.
So the next question we have is there a reason why we look for frequent patterns or strings?
Right. So as I mentioned, I did not go into the biology details of it. So what happens is that, say you're looking at a replication origin gene, right? The sequence. There has particular patterns which have been repeated quite a lot of times, right? Say ATGC. So if you look at one particular string, the chances of it being A is only 25%. The chances of it being C is only 25%, right? And if you look at two, the chances of being it AT is going to reduce by 12.5%, right? And if there is a huge sequence, that being repeated is extremely low, right? Just randomly getting repeated. So if it is getting repeated more often, that essentially means that that particular sequence is a coding region, or it's a region where the DNA A boxes come and bind to it. So that way we can look into the biological questions of uh, of the genes or genetic therapy or whatever. Yeah.
Okay.
Yeah.
So the next question we have is, when you use frequent string, is there a way to further reduce the list?
Yes, there definitely is further to reduce the list, and it's a very important question. I do have a slide for it, right? So I didn't go into the details of the DNA because I don't have much background in biology. So if you look at DNA is a double helix model. So A is adenine, which always binds with T, and G binds with C, right? So if you have a ATGC, that means that you can, if you have one string of the double helix model, you can easily replicate the mirror image. So the second part of the string is just the mirror image. So one thing is, if say in this one, we got TTGAGCAC and again AATCGTA. So that's the mirror image of each other. So then we can just remove the mirror image from the frequently repeated strings.
Okay.
I hope that makes sense.
Yeah. So I had a question of my own, just one final question. So which is so what are some of the advanced machine learning like, because so one of the advantages of okay, we have before I
Another
go into
question.
the question. First of all, your coding was so neat. I have a question regarding gene expression data. I'm studying gene regulatory networks inference from dynamic gene expression data. I'm writing an optimization algorithm for the inference based on S system equation. Could you suggest any source or link for any Python library that can be used specifically for bioinformatics gene regulatory networks inference from the expression data?
Right. So I don't have any link at the top of my head, but I can look into it. I can share my email ID with the student, or you can. I think you can also have my email ID is also provided in the flyer. So
Sure.
if you send me an email,
Uh,
I will get back to you.
while updating the slides and the video recording, you can share the
Right.
links with
Yeah.
us. Thank you.
Yeah.
So yeah, just on those lines, yeah. So just on those lines, what are some of the, because one of the advantages I sort of personally see with Python is that you've got a lot of open source libraries where for some particular type of work you have a dedicated library, so which you can use, like just similar to how we got the previous question. So what are some of the, say, advanced machine learning libraries that you can use for bioinformatics? Like, yeah, any machine learning libraries in particular, or the regular ones like sklearn have got features?
Um, Well, as I said, like I've just started this bioinformatics field. I'm looking into it, and the things are it with machine learning. It depends on the type of the problem and the goal of the problem. So it varies. But I have seen people using PyTorch, Keras, and in one of the basic one which course I did, they use this teapot library, which basically searches all the important machine learning libraries depending on the data structures you have, and it gives the optimal machine learning algorithm. So yeah, I think it mainly depends on the type of problem that you have.
Thank you so much, everyone. Thank you so much, Sanjay. Thanks, everyone. So that brings us to the conclusion of the talk, and that brings us to the conclusion of our symposium talks. So we had a range of talks spanning from, like, I can't even recall most of them now because we just had such diversity and such high quality talks that span different topics. So tomorrow we will be having our closing ceremony. That is Thursday, 3rd December at 5 p.m. So we'll just discuss few things and whatever resources we can share, and then some of the computations that we had for the data analytics things, just announcing prizes for that and all those things. So we are all looking forward to seeing you one last time tomorrow at 5 p.m. Until then, stay safe. See you. Thank you, Sanjay, again.
Thanks.